Hello everyone. Hello to all the subscribers of Biotechnica. This is Tithi here and welcome to another video where we are going to talk about something important for your exam preparation. So today's video is on advanced topics in molecular biology. And in this video, the advanced topics for molecular biology that we are going to cover is not only for CSIR NET Life Science preparation, exam preparation, but as well as that is going to help you for your GATE PT 2025 exam preparation as well. So stay with me till the end. So what is the importance of molecular biology in CSIR NET as well as GATE? Okay, so for that matter, if I say for any competitive exam, molecular biology, like any competitive exam related to your PhD admission in life sciences, molecular biology is an extremely important segment. Now, this is the part, one of those part of the syllabus which you cannot ignore at all. And also, whether you are from pure sciences, pure life science, or you are from applied life sciences, like we all know that life science is very broad and there are many subdivisions of life sciences. So whether you are from pure or applied life sciences, you are bound to know the basics of molecular biology. The reason being that what is molecular biology? So molecular biology basically comprises the way DNA, which is our genetic material, is converted into RNA, and how that RNA, how the information in the RNA is read to convert it into protein, okay, and also how this DNA is self-replicated as well. This all processes, which is this one, is cell DNA replication. The process of conversion of DNA to RNA is transcription. RNA to protein is translation. So all of this together is what we call as the central dogma. And the ways in which we study the central dogma and we find out the different ways by which we can modify these different processes, that is what is being the part of molecular biology. So, and because these are the processes which has to happen in all the cells, it is required for the cells to grow, to reproduce, to survive. So that's why they are very truly known as the fundamental processes. In fact, in CSIR NET syllabus, they are covered under fundamental processes of Unit 8. Although molecular biology, because it is so vast, again cannot be confined into one unit, and there are different portions of molecular biology which comes in different units as well. Okay, so that's why if you remember, if you attended my previous videos, you might have noticed that I always say that when you are preparing for any exam, you always have to see the bigger picture. These are all, like, the syllabus is divided into different topics, but these topics are connected because anyways we are talking about most of the things which are happening inside the cell. So all of these things are kind of connected, and you should have a basic understanding of this connection. Then only you will be able to answer the questions which come in this in the exams. So from CSIR NET syllabus, this is Unit 3, but yes, the topics are covered in different other units as well. From GATE PT syllabus, it is Section 3. In Section 3, there is molecular biology, but again, it is correlated with cell biology. It is correlated with genetics as well. So when we are studying molecular biology, what kind of understanding you should have? So because it is again so vast, so your concepts, your basic roots of the knowledge should be completely clear. So it is important for understanding the fundamental process of life at a molecular level. Now for excelling this particular topic, what you can do is you can stay updated with recent research, like whatever there is in the standard reference books. Definitely they are good and you have to learn them. You have to have the conceptual understanding of them, but along with that you also should be aware with the recent research which is going on in this field. Especially this is again very much important if you are applying for CSIR NET exam, because there 1 or 2 exam might be part B or part C comes from the recent research which is going on molecular biology. And when you are preparing for it, so it enhances the conceptual understanding and the application skill as well. Now, what are the list of advanced topics which are covered under this, or in molecular biology, what advanced topics come? So very first thing is DNA replication. Now, when we talk about DNA replication, yes, you know, it is the process of how DNA is replicated, but it is not confined to that, because here we also have to talk about DNA repair and recombination. Now, repair and recombination are two important aspects which make sure that our genetic material remains unmutated or unchanged. And if there is any fault, or if there is any if the repair and recombination process is not working properly, that is when changes will happen in the DNA, and definitely that will have different sort of implications, which we'll cover in another video maybe, where we will talk about the various mutations. Yeah, so after DNA replication, the next advanced topic will be RNA processing. Now, when we are talking about RNA processing, so what all things? So we'll see it in detail as well. So let me give you a brief idea. So RNA processing basically will comprise of all the post-transcriptional modifications which are happening in the RNA, because those processes are like transcription, which is conversion of DNA to RNA, is happening inside the nucleus, right, for the eukaryotes. But that RNA has to be transferred to the cytoplasm to do rest of its job, and because of that, the RNA has to be modified so that it sustains, or it becomes stable, or it remains stable in the cytoplasm. So all those post-transcriptional modifications, which is an important aspect of eukaryotic transcription, is a topic that is very important from molecular biology. Next will be gene expression and regulation. See, we know that DNA has, the gene will be present in the DNA, and gene will code for a protein. But just having a gene is not enough, or it doesn't ensure that the cell will have its expression also. Expression means the protein that is coded by that particular gene. So gene expression and regulation, so at various levels, the gene expression is regulated to make sure that there is no wastage of any product, there is no wastage of energy, and the optimal amount of product is produced at the correct time, at the correct place inside the cell. Next is protein structure and function. So we are going to talk about the various ways the protein structure affects its function, and the various modification which happens in protein to give it the stability, and then the various techniques in molecular biology. So I will give you a brief idea, a little bit more detail we'll talk about each of these advanced topics. Starting with DNA replication, repair and recombination. So from exam point of view, GATE as well as CSIR NET, so what all things you should first of all master from this? Okay, so first of all, you should have a very good understanding of the basic mechanism of DNA replication. Means you should know the semi-conservative replication. You should also have a very good understanding of Michelson and Stahl experiment. So although that experiment happened so many decades ago, but still, because that was such an important step in understanding DNA replication, that questions do come from there. Okay, so Michelson and Stahl experiment to show the semi-conservative mode of replication is something that you have to study very in depth. Then role of DNA polymerases, helicases, primases, ligases. So basically all the enzymes which participate in DNA replication, you should have a understanding of their roles, and also single cell binding proteins. Now along with that, something I would add is also the role of topoisomerases. Okay, whether it is topoisomerase 1, topoisomerase 2, how they help in reducing the supercoiling, what increases the supercoiling. Okay, so you should have a good understanding of that and the work and mechanism of topoisomerases. Next one is about replication, origin, and licensing. That is the regulation of DNA replication in eukaryotes as well as in prokaryotes. You should have a good understanding of that. And moving on lastly to DNA repair mechanisms. So all the DNA repair mechanisms you should be completely thorough in. Whether it is base excision repair, nucleotide excision repair, mismatch repair, or double strand break repair mechanism, which we also call as homologous recombination, as well as the non-homologous end joining, which we call as NHEJ pathway. Okay, so and all of this are various different types of DNA repair mechanisms which happens depending on what kind of change has happened in the DNA. See, when we talk about mutation as such, okay, so definitely we know that there are chemical agents or physical agents which can cause mutation, which are called as mutagens. But mutation are not only happening when there is mutation, when there are mutagens. Mutation happens also when there are no mutagen. Okay, so basically, and those kind of mutations are called as spontaneous mutations. And how do they happen? If there is any fault in the DNA repair mechanism, that is when the mutations happen. So that's why the cell will try to make the DNA as it is, keep the DNA as it is, because it is so important. It codes for the basic it is the blueprint of the life for the cell, right? So it doesn't want any change in it. So that's why all these DNA repair mechanisms are there to correct any mutations. All right. So only those mutations or those alterations in the DNA which escapes this DNA repair mechanism, they only stays in the DNA, or they only cause the various evolutionary changes that might have happened in the species across this millions of years. Now, how to study this DNA repair mechanism? Yes, there are different DNA repair mechanisms. It's very easy to confuse between them. So the best way what you can do is you can make a comparison table. Okay, so you can make a comparison table with all 2 or 3 or 4 different DNA repair mechanisms on one page. Okay, so and write them all together. So everything you will have it on one page, and it will be excellent, and write only the important things, and it will be excellent for your revision. Next topic will be RNA processing and post-transcriptional regulation. Now, in that, when we talk about RNA processing, you should be thorough with various RNA splicing and RNA editing mechanisms. Questions very frequently has come. Now, this too, like RNA splicing and editing, questions come very frequently in case of CSIR NET. Okay, these two topics are extremely important for CSIR NET. mRNA transport and localization, it is kind of important for both. Okay, NET plus GATE. And RNA interference is something which is very frequently asked in GATE. Now, in RNA interference, basically what we are going to talk about. So, an RNA interference can be included in RNA processing, or it can also be included as a part of gene regulation as well. So basically, where the cell using miRNA, that is microRNA or siRNA, it controls the expression of different genes, either by marking the protein coded by that particular gene for degradation, or by making the mRNA coded by that gene double-stranded, so that the mRNA cannot be taken for translation. So basically, this RNA interference works in silencing the gene expression. So that is why the gene will be there in the cell, but still the protein expressed by that gene will not be present. Okay, so there are many cells in our body, but at a single time, if you see, only 10% of those genes only are expressed. Okay, so there are different regulations, different gene regulations that are happening in the cell simultaneously to make sure that there is optimum usage of resources and optimum amount of only those products which are needed by the cell at that time is produced. So when we come to gene expression, so then the next important topic, gene expression and regulation. So here you should have a good understanding of prokaryotic regulation as well as eukaryotic regulation. So in prokaryotic regulation, there is operon structure. Definitely you cannot escape that, and it will be especially you should have a good understanding of lac operon and tryptophan operon, various transcription factors, enhancers, silencers, promoters, etc. in eukaryotes. Then you should have a good understanding of eukaryotic gene regulation as well, which comprises of the various chromatin structure and remodeling. So basically in the eukaryotes, what happens is the DNA is tightly wrapped around it is tightly wrapped around the nucleosome, which is comprising of eight histone molecules. Okay, so 2 of each kind, and histone modifications can happen, that like acetylation, methylation, phosphorylation. So basically various amino acids in different positions of the histone molecule can get can be acetylated, methylated or phosphorylated, and that will change the way the histone is interacting with the DNA around it. So some modification will make the DNA tightly wrapping around it, and if the DNA is tightly wrapped, the transcription machinery will not be able to reach the DNA and the expression of the DNA will be stopped. But at the same time, if any modification is making the DNA go loosen around the nucleosome, in that case, the expression of the gene will become easier. So in fact, when we see those genes like the housekeeping genes, right, the genes which are always required by the cell, so housekeeping genes occupy those segments of the genome which are not at all wrapped around any histone molecule. Okay, it will be completely loose, so that transcription machinery can always access it and convert it into the product. Then you should have an understanding of epigenetics. So epigenetics is the control which comes over genetics, and that happens by DNA methylation, histone code hypothesis, and also the various non-coding RNAs. So as I told you, siRNA, miRNA, their interference can also be counted here as well. Okay, so these are the things that you should know for gene expression and regulation, and yes, it is one of the important topic for both CSIR as well as GATE. Then protein synthesis and post-translational modifications. So here you have to have an idea about translation, how basically the translation happens. Especially you should know about the genetic code or the codons, role of different tRNAs, role of the tRNA, structure of tRNA is something which is extremely important, and also ribosome structure and function. Along with the various steps of translation. Okay, although questions don't come exactly regarding the steps, but yes, question can come where you have to correlate an experimental situation or an analytical situation with the basic understanding of the processes of translation. Okay, like in case of prokaryote, so what are the differences in prokaryotic and eukaryotic translation? So in case of prokaryotic translation, either it is mRNA or mRNA binding with the small ribosomal subunit, or the initiating tRNA coming to the small ribosomal subunit. It can happen in either way. Okay, but when we talk about eukaryotic translation, there is a specific way in which binding happens. So it will always be the initiating tRNA which will first come and bind with the small ribosomal subunit in eukaryotes, and then only the mRNA will be brought to this smaller subunit. So these are the differences, and so basically there can be questions where you have to apply this basic knowledge of translation. Now definitely nobody will ask you to write a passage on translation, nobody will ask you to describe the different steps of translation, but these are the questions can come in such a way that you have to apply this basic knowledge. So definitely you should have an understanding of the mechanism of translation. And also the various post-translational modifications, because when translation is done, it just produces a string of amino acids, but that string of amino acids have to be modeled, have to be molded into different forms to form a protein that is functional, and for that various other modifications happen: phosphorylation, glycosylation, ubiquitination, protein folding as well as degradation, along with that carboxylation, glycosylation. So these are the various different processes that have various different chemical modification which happens in different amino acids in that polypeptide chain to convert that polypeptide chain into a functional protein. So you have to focus on this post-translational modifications also. Coming to the molecular techniques now definitely for a molecular biology will be incomplete if we do not include the molecular techniques. These molecular techniques are part of Unit 13 in CSIR NET syllabus, and it is part of Section 7 in GATE syllabus. Okay, so whether you're talking about CRISPR-Cas9 technology, which is one of the very recent and modern technology where a lot of molecular biological work is going on, so you should know that. Next generation sequencing along with the basic sequencing also, Sanger sequencing, determinator method, pyro sequencing, this like principal techniques and applications along with the various next generation sequencings you should know. PCR and qPCR, some of the very basic molecular biology technique that happens in all mol bio labs. Okay, so you should have the understanding of that, and also various molecular cloning and recombinant DNA technology. So all the students, if you prepare all these 5 topics in detail, and as well as how I said, right, different topics from different subtopics from all these topics, if you prepare that, your base for molecular biology will be strong and you will be able to solve any question that comes from this section, whether it is CSIR NET or GATE, BT or Excel. Thank you everyone for watching. We will come again with some another important and helpful video. Till then, bye-bye. Thank you everyone.